Hello, my name is John Pennant and I'm a mathematician specialising in partial differential equations, and I'd like to talk about an engineering problem which I solved working on the Thrive project. The Thrive project is an attempt to create an open source computer game which creates high impact science communication. So a bit like Kerbal Space Program did for rocket science, it gave you a sandbox where you could add all these different components to your rocket and then fly it into space and see how it acted. We're trying to do something a bit similar but for cell biology at the moment. And just like the human body has organs inside it, so heart and lungs and liver, etc., a cell, an animal cell, has organelles inside it, like a nucleus or a Golgi body or a mitochondria. And yeah, so we're making this game where you can add these different organelles to your cell, and then you can swim around, you can fight other creatures, and you can evolve. And it's meant to be kind of a game of interest to people who are interested in evolutionary game theory or microbiology, who like the scientifically accurate side. And it's also meant to just be fun. It's meant to be a cool thing, like Kerbal Space Program, just fun to get in, play around, and have a good time with. So it's an awesome project. If you're interested, check it out. I'll put a link in the description. And I want to thank all the other people who work on the Thrive project. You guys are awesome, and it's been a real pleasure working on it. So I want to talk about a specific problem that I was working on, and I want to thank the other guys on the team for helping me with it. Basically, imagine you have a cells, for example, like this amoeba. It has a cell membrane. It has a lipid bilayer, so it's sort of fats, which are two layers thick, going around the edge. And it needs a cell membrane because it's sort of saltier on the inside of the cell than it is on the outside. So there's this continuous osmotic pressure of water trying to cross over this membrane and equalize the pressure, equalize the salinity. And so it needs to have a membrane which holds the nice tasty things inside, all its sugars and proteins and that kind of thing. So how computationally are we going to model this membrane? And the constraints are, we want a relatively scientifically accurate description of a membrane surrounding this cell, but we also need it to be computationally cheap. So if we've got players who are playing on older computers, we need them to have to be able to compute this membrane very cheaply. So what did we do? So we played around with a few ideas, and the solution we came up with was, imagine you have a nucleus of a cell, and then you put lots of spikes coming out of this nucleus. Okay, so you can see in this diagram is in 2D, but the real solution was in 3D. But I've done it in 2D just to make the diagram easier. So you can see on the left-hand side there's a diagram, and the thetas, theta 1, theta 2, theta 3, are the angles at which these spikes leave the nucleus. And these are fixed, these will never change. And when it's in 3D, you get two angles, because it's spherical polar coordinates, so you get a theta and a phi, say. But for this 2D, it's just that. And you also get a radius, so R1 is the radius of this first spike, and R2 is the radius of the second spike, and R3 is the radius of the third spike. And it's the points on the end of the spike which determine the edge of the membrane. So, because these spikes never move, they just move in and out, you can sort of imagine this spiky ball with all these things coming out of it, defining this membrane which covers the cell. So how should the points at the end of these spikes move? The spikes aren't real, they're just to define vectors defining the positions of the points. So how should they move? Well, the first thing we want is for the membrane to be smooth, and so the idea I came up with was to use the heat equation. So you can see the heat equation on the right, dTRI equals Laplacian Ri. So what you want, this is a very good equation for this because it creates C infinity smoothness in very fast time. That's a technical PDE thing. But what it's doing is it's smoothing out the membrane. So you can see above the discretized Laplacian for R2. So R2 is one of these spikes, and it's the distance R2 from the nucleus. And what it does in 2D is it takes the values of its neighbours minus 2 times the value of itself, and that's the value of the Laplacian at that point. So imagine if R1 was equal to R3, then what R2 is trying to do, what the Laplacian is trying to do, is to get R2 to be the same length. It's trying to equalise these lengths. So if R2 was greater than R1, which was equal to R3, then the Laplacian would be negative. And if R2 was less than R1, which was equal to R3, the Laplacian would be positive. And so what this means is that R2 will move, using the heat equation, to create this smoothness, to average out the distances around this whole sphere. And this is really cool. So when you just create the model with just this heat equation on it, it goes from whatever initial conditions you pick, maybe they're very spiky, it will then smooth the whole sphere to be a sphere. It will smooth it towards being a sphere. And this is really cool because it gives us this lovely smoothing property which makes it look very organic. Okay, but it's not really enough because we want to have the organelles move around, and that it doesn't look like the amoeba which we were modelling. Okay, so an extension to this is, now if you see it, look in the diagram on the left, that blue circle is now an organelle, so the black circle is a nucleus, and the blue one, maybe it's a mitochondria. And so for each point, you find the minimum distance, you find the distance to the closest organelle. So for example, for R1, that distance is D1, which goes to the closest organelle. Okay, and for R2 it's D2, and maybe for the other ones there's other organelles which are in the cell as well, but you find the closest distance. And then we change the formula to be dTRI. So how does Ri change in time? Well, it's the Laplacian of Ri, which is going to smooth it, plus this second term, alpha e to the power minus beta di. Okay, and what this means is as di approaches 0, so as, for example, for R1, if d1 starts to become small and it starts to get close to this organelle, this term will start to become large, because as di gets smaller, e to the minus beta di will get larger. So depending on the values you pick for alpha and beta, and they're just scaling which we set cosmetically, this term will be positive when you get too close to an organelle. So it will tend to push the membrane away. So if the membrane's being smoothed out, but it's coming too close to an organelle, then it will be pushed away. And gamma, then, finally, is a shrink-wrapping term. So technically, if you just have this exponential term, the membrane will technically grow forever. It will grow slower and slower the bigger it gets, but e to the minus beta di will always be positive, because di is always positive. And so, well, in fact, e to the anything is always positive. So the gamma term shrink wraps the whole membrane around the organelle. So you want it tightly around the organelles. So if minus gamma was the only term, all of these points would shrink down and just wrap very tightly around the nucleus. So with the three terms between them, the exponential term is pushing them away from the organelles, the gamma term is pulling towards the centre, and the Laplacian term is smoothing out all of these points. Okay, so you've got these three different forces going on. And you can see in this video how that looks in the prototype. So we start out with a cube. We start with all the grid points set out in a cube because that was very easy to parameterize. And then when we turn the model on, you can see it smoothly wraps around the organelles, and the membrane moves in a very nice organic looking way. And if we allow the organelles inside the membrane to move, then the membrane will move with it and it will completely encapsulate it. So you can see how this is quite nice. It really does look like, relatively like the amoeba which we were modelling in the first place, and it has a nice smooth organic motion using these differential equations techniques. And so now here is a video of the membrane actually in the game Thrive itself, done by the brilliant Michael Silva. He's our graphics team lead and he implemented this in the game. And as you can see, it looks pretty cool. The implementation is slightly different from the prototype. But yeah, it's I think successful and makes it look like a real microbe. So I hope that's been interesting. I hope I've explained it well. Basically, the idea is, how do you apply abstract PDE techniques? Well, this is an example of applying abstract PDE techniques to a specific problem and constructing a differential equation in such a way that you get the dynamics you want. Often PDE study is built around you given a differential equation, like say Navier-Stokes, and you're asked to say what kind of dynamics will this create. In this way of working, you think about what kind of dynamics you want the equation to have, and then from that you construct the equation which will give you these dynamics. So I hope that's cool, I hope that's interesting, and it was a pretty cool problem to work on, and I think working on Thrive is brilliant in general. So Thrive is open source, and we're always looking for more contributors, so if you're a programmer, if you can do graphics, if you're a musician or an artist, or you're a theorist, so you've got scientific knowledge either in biology or mathematics or anything else, chemistry is fine, come along, find our website, link in the description, and join the team and help out, because it's a really cool project, it's a really cool thing to be doing, and this is a cool problem to work on. So I hope this has been interesting. I'm John Pennant. Thank you very much for listening to PDE's talk. I appreciate it. Thank you. Goodbye.